Cortisol. Is it really just the stress hormone? What is the first thing you think about when you hear the term cortisol? Anxiety, stress, fight or flight perhaps? Whilst these are definite responses to the hormone cortisol, it is a complex and important hormone which has a cascade effect throughout the body, responsible for many functions. Cortisol is intriguing yet complex, so let's get started and try not to stress out. Also known as hydrocortisone, the term cortisol is derived from the Latin word cortex, meaning cork tree, essentially the outer shell of something needing protection. Hormones are chemical messengers controlled by the endocrine system. The intricate biochemistry of the body relies on millions of reactions happening at any given time. From tasks like triggering hunger or complex functions of reproduction, all are governed by these messengers. Classified into three groups, amines, proteins and peptides, and steroids, cortisol, a principal glucocorticoid, is a sugar steroid secreted by the adrenal cortex along with the principal mineralocorticoid, aldosterone. Steroids, due to their lipid-soluble nature, cross the plasma membrane easily, beginning protein production at the nucleus. Adrenals, or suprarenals, sit superiorly on the kidneys with a weight of 4 grams. Of all the organs in the body per gram of tissue, the adrenals have the greatest blood supply, each being serviced by up to 60 small arteries. Cortisol is made by the hypothalamus and secreted by adrenocorticotropic hormone, ACTH, from the anterior portion of the pituitary, one of 10 glands comprising the endocrine system. An antagonist to insulin, it aims to increase blood sugar levels when they dip. ACTH is a large polypeptide and, like other pituitary hormones, is controlled by a releasing factor of the hypothalamus called corticotropin releasing factor and is in charge of releasing cortisol in a stress response. Comprised of epithelial cells, the adrenal cortex consists of the deep adrenal medulla and 3 superficial zones. The outermost region, the zona glomerulosa, is responsible for mineralocorticoids, with secretion being regulated by cortisol and androgens produced in the second and third layers of the cortex. Glomerulosa is the thinnest of the layers, while the zona fasciculata is the thickest. Here is where cortisol and other glucocorticoids, such as corticosterone, are secreted. This secretion is predominantly controlled by the HPA axis, hypothalamic pituitary adrenal axis. The third innermost, bordering the adrenal medulla, is the zona reticularis, named due to its cell arrangement in a reticulum network fashion. It is responsible for producing androgens such as dehydroepiandrosterone and testosterone. Glucocorticoid synthesis is stimulated by ACTH, which is released in the bloodstream by the anterior pituitary. The axis is an example of a negative feedback system in the body, characterized by cortisone acting as an inhibitor of ACTH and CRH. Metabolism is a multifaceted process utilized by all living organisms. Glucocorticoids are just part of a big picture in humans and are primarily responsible for circulating glucose due to mobilization of amino acids from protein sources. Regarding carbohydrate metabolism, glucose is synthesized from amino acids in hepatic tissues when blood glucose is low. This metabolic gluconeogenesis pathway occurs when the liver converts proteins and glycerol portions of fat into usable glucose. When typical levels of carbohydrate become unavailable to hungry cells, the anterior pituitary begins secreting corticotropin, then stimulating the adrenal cortex and significant quantities of glucocorticoid hormones. Gluconeogenesis is essentially the reverse process of glycolysis, where glucose is broken down to produce pyruvate from the cytosol into the mitochondria and then on to become acetylcoenzyme A, due to the pyruvate dehydrogenase enzyme. Gluconeogenesis is similar, except for a sub-step at the beginning, kicking off the conversion of pyruvate towards glucose, regulated by acetyl-CoA. This step is unidirectional and cannot be reversed. If complete reversal of glycolysis occurred, energy expenditure would be too great for it to be profitable for the demanding organism. The enzymes of glycolysis and gluconeogenesis in the liver are reciprocally regulated so that either glucose is converted to pyruvate or vice versa. Our 24-hour internal clock and specific stimuli are the 2 incentives for glucocorticoid secretion. Specific stimuli are triggers that send nerve impulses to the hypothalamus, activating the sympathetic portion of the autonomic nervous system and increasing adrenal secretions. Whilst cortisol has positive effects on the body and its desire for a homeostatic state, like waking up in the morning, it also has negative impacts too. When high levels are in circulation, glucose utilization decreases in the majority of cells. Whilst the mechanism is unclear, physiologists hypothesize that a truncation of the process of glucose entering the cells occurs, resulting in a delay of uptake and use. Endurance exercise events, a keen hobby of mine, and its benefits and harms to the physical and psychological components of our being, has found many points of contention. Research in this area is beneficial for athletes at both the elite level and the layperson enjoying sport for health and well-being in their community. Whilst releasing cortisol under threat in the environment thousands of years ago was appropriate, it is not uncommon to see bodies being pushed beyond healthy boundaries in modern society. Overtraining syndrome is prevalent in sporting arenas, resulting in a plethora of issues, both short and long term. In this paper from 2000, Lucille Smith analyses the area of overtraining and adaptive capacity. Smith states, with educated progressive training increases, the HPA axis should stabilise, i.e. adapt. However, with excessive physiological and/or psychological stress, delicate hormones can be disrupted. Seconding this, half a century prior, was Austro-Hungarian endocrinologist Hans Selye, who in 1950 developed the stress theory, observing that countless pathologies were derived from hyperinflammation or immunosuppression within the body. Pathological progression occurred in 3 stages: alarm, resistance, and exhaustion, and coining the term general adaptation syndrome. With inflammation within tissues, cortisol increases the struggle for intracellular lysosome membranes to rupture, causing a decrease in quantity and efficacy of proteolytic enzyme activity. In this state, the pancreas produces glucagon, and with the anterior pituitary secretes growth hormone, forms energy sources, and stimulates the absorption of amino acids to aid in tissue repair. Like Smith, Selye identified the alarm and resistance stages had the propensity to be adaptive opportunities, and the exhaustive stage was organisms being depleted of its reserves, overtraining. 5 stages of inflammation were noted. Firstly, histamine, proteolytic enzymes, prostaglandins, and leukotrienes are released. Secondly, vasodilation occurs to allow fresh blood to nurture the tissues. Thirdly, increased capillary permeability and quantities of plasma to flush out the area. Fourthly, infiltration by leukocytes. And fifthly, fibrous tissue regrowth. Perhaps with graded approaches, guided levels of oxidative stress in order for the present free radicals to undergo repair is possible. Combined with antioxidants such as amino acid-rich glutathione, the load the mitochondria of our cells can withstand could increase. Researchers looked at links between intense long-durational physical activity and its connections to increased blood cortisol levels, decreased free testosterone, and connections to systemic inflammation. Research into overtraining, such as the female athletic triad, looks closely at reproductive implications, such as testosterone reduction being incriminated as affecting the HP gonadal axis, and thus the controlling hormone of GRH, which triggers the release of gonadal, luteinizing, and follicle-stimulating hormones. Consequently, ovarian estradiol and testicular testosterone are deployed. These inflammatory mediators suppress reproductive function via inhibition of GnRH. Other repercussions include osteoblastic capacity obstruction, GI tract calcium absorption decline, timing of birth in pregnancy, and in the area of mental capacities, cognitive impairment, memory retention, hippocampus decay, and mental illnesses such as depression and anxiety are all connected to levels and length of exposure to cortisol.